Then chromatography. The chromatography, that is the collective term for the set of the laboratory techniques for the separation of mixture. The term comes from the Greek word chroma, that means the color, and graphene, that means to write. So the literally meaning of the chromatography is the color writing, because it was first used in the mid 19th century for the separation of the color pigments like chlorophyll. For the separation of analyte substance, the mixture to be separated is dissolved in a fluid called as the mobile phase, which carries it through the another material called the stationary phase. Various constituents of the mixture travel at a different speed, causing them to separate. The separation is based on the different adhering powers between the components of the mixture and the stationary phase. Components which adhere weakly to the stationary phase move the faster, and those that adhere strongly move slower. Suppose a number of men, women, children are wading through a waist high water with a sticky mud at the bottom. Although all start together, but after some times they would separate depending on their individual strength. Same happens with the molecules in case of the chromatographic plate, and this depends on the R value, that is rate of flow. As the solvent, that is the mobile phase, pushes the components of the mixture stationary phase, they move differentially depending upon their affinity to the stationary phase. The rate of flow, R value of the solute, is more if it flows farther. RF, that is rate of flow, is the distance from the baseline traveled by the solute divided by the distance from the baseline traveled by the solvent. As you can see in the picture, this is the chromatographic plate. Here at a point the solvent of the interest is put and it is dipped into the mobile phase. And this on this chromatographic plate, a stationary plate is there. On chromatographic plate, a stationary phase is there. So once this is dipped in a mobile phase, different particles in this solution will travel in the different speed according to their affinity to this stationary phase. So this mobile phase will allow the different molecules to travel at different speed, and here they will produce the different colors according to their positions. This will be detected to know the exact nature of the substance. So this is the idea behind the chromatography. So here RF value are best illustrated by the paper chromatography. That is the RF value, that is from here the solute that is taking up to here, this distance divided by the solvent that is taking to the maximum distance. So that is the RF value, that is rate of flow. So each colored dot represents a different compounds. As solvent rises up, chemicals travel along with it. Their speed of travel vary according to their affinity to the paper and the solvent. This causes separation of each chemical. R value don't have units since it is a ratio. Every compound has a specific R value for every specific solvent and solvent concentration. Compound will have different R value for the different solvents, and by this we can identify the exact compound. Then chromatography is classified depending upon different conditions, like depending upon its purpose, preparative chromatography, analytical chromatography, depending on the shape of the stationary phase, that can be the column chromatography, planar chromatography, that includes the paper chromatography and the thin layer chromatography, depending upon the physical state of the mobile phase, that can be gas chromatography, liquid chromatography, HPLC, that is high pressure liquid chromatography, and adsorption chromatography. The preparative chromatography used to separate the components of the mixture for further use. It is a form of purification. Large amount of the mixtures may be used at the preparatory form of the chromatography. Then analytical chromatography is done with the smaller amount of the material and used for measuring the relative proportion of the various components in the mixture. This form is mostly used in forensic laboratories. So preparatory chromatography that we discussed while extraction techniques. So there it is preparatory chromatography, and when it is used for analytical purpose, then it becomes the analytical chromatography. Then depending on the shape of the stationary phase, that is that chromatographic plate, that what type of chromatographic plate is being used, that chromatography is the type. So, in the column chromatography, a stationary bed is the shape of the column within the tube, and in planar chromatography, the stationary bed is in form of a plane like the paper chromatography. If the plane is paper either as such or impregnated by a substance, then this is called paper chromatography. Then in thin layer chromatography, if the plane is in the form of thin layer of a solid particle sprayed on a support such as glass plate, then it is called thin layer chromatography. Then depending on the physical state of the mobile phase, the gas chromatography, in which the mobile phase is in the gaseous state, and in the liquid chromatography the mobile phase is the liquid. When a high pressure is used to force the liquid through the stationary phase, it is called the high pressure liquid chromatography. And if the mobile phase is gas or liquid that is adsorbed onto the surface of the stationary solid phase, it is the gas solid or the liquid solid chromatography. That is a type of the adsorption chromatography. As the separation occurs due to differential adsorption of the compounds on the solid stationary phase, it is also known as adsorption chromatography. Adsorption chromatography is the one of the oldest type of chromatographic method. Then coming to the individual chromatographic methods. The column chromatography, that is used for both preparative and the analytical chromatographic purpose. Its advantage is that it is low in cost and disposability of a stationary phase is used in the process, so it prevents the cross contamination and stationary phase degradation due to recycling. So here you can see that stationary phase is in form of the column. So here you can see the stationary phase is in form of the column. A sample is loaded on the column and then the mobile phase is added, so that it will cause the movement of the constituent solutes of the sample on the column. So different constituent will have the different moment. If it is having a stronger interaction, so it will move the lesser, and weaker interaction will move the farther. So in this way we can also extract the different compounds by further taking it down, and likewise eluted molecule of different categories are extracted. So it is getting used for both the preparative phase and the analytical phase. In this way column chromatography works. Then another technique is the paper chromatography. Paper chromatography is an analytical technique for separating and identifying mixtures that are or can be colored like pigments. It has been largely replaced by the thin layer chromatography and is currently used mainly as a powerful teaching tool. Two-way paper chromatography, also called two-dimensional chromatography, involves using two solvent and rotating the paper 90 degrees in between. This is useful for separating complex mixture of the similar compounds like those of the amino acids. So here you can see in the earlier example there also this paper chromatograph that is used, it has the solution is kept on this line and this is put in the mobile medium, and there this will travel, mobile phase will travel on this stationary medium, and that will take the different solutes along with it, and depending upon their distance we can judge what type of compound this mixture contains. So by this method paper chromatography is applied. Then the thin layer chromatography. Thin layer chromatography is similar to the paper chromatography, except that instead of the paper it is performed on a sheet of a glass, plastic or the aluminum foil, which is coated with the thin layer of the adsorbent material, usually silica gel, aluminum oxide or cellulose, or that is on the blotter paper. This is the simplest analytical technique commonly used for qualitative screening of large batches of the samples employed in many poisons like that of organophosphates. So you can see here in this picture that is depicting the performance of the thin layer chromatography, that is showing the different types of the organophosphate compound that will have the different distance travel as well as the different colors that can help to detect the particular type of organophosphate compound. Then the gas chromatography. In gas chromatography, the mobile phase is the carrier gas, usually inert or the unreactive gases like the helium or the nitrogen. The stationary phase is the microscopic layer of the liquid or the polymer on an inert solid support. here in the gas chromatography, the stationary phase is the microscopic layer of the liquid or the polymer on an inert solid support inside a piece of glass tubing called the column. The instrument is called a gas chromatograph. The compound to be analyzed is vaporized into the gas, which is pushed in a system along with the carrier gas. The particles interact with the stationary phase, and depending on their mutual affinity, the particles come out of the system at the different times. The time for which a compound is retained inside the system is known as the retention time of the compound. The analysis of the retention times reveals the nature of the compound. Since in this system, the compound equilibrates for the partitions between the mobile gaseous phases and the stationary liquid phase. It is also known as the partition chromatography. So you can see in the picture, this is a gas cylinder containing the inert carrier gas that acts as a mobile phase. So once through this inlet, the mixture of our interest is injected. This gas cylinder act as a mobile phase and causes the mixture to pass through this column, and in this column different adsorbents are there, usually silica gel, so that the affinity of the different constituent of this mixture will have different exiting speed from this column. So the detector will detect which substance has come out at what time and what is the, according to that they will create a chromatograph, and that is can be used to detect the type of the nature of this mixture. So by this, gas chromatography is used. Then another variant or the adjuvant of the gas chromatography is the gas chromatography and the mass spectrophotometry. As I told you, this combination is considered to be the gold standard, as it is 99% sensitive and specific in detection of the poisons. So under this is the lab picture of the GCMS, and here after the performance of the gas chromatography, the ejected material or the constituent is subjected for the mass spectrophotometry, and they will, according to the mass charge ratio, they will travel through the mass spectrophotometer and they will be detected on the chromatograph, and that will help to decide the type and the nature of the compound. So in this way, this GCMS is functioning. Then the high pressure liquid chromatography. High pressure liquid chromatography is similar to the gas chromatography, except that it is not restricted to the volatile compounds. Here the mobile phase is drawn from a reservoir into a pump, which controls the flow rate and generates the sufficient pressure around 100 to 6000 pounds per square inch to drive the mobile phase through the column. An injector or auto sampler is used to place the sample on the column without stopping the pump's flow. Multiple samples can be injected in a turn by turn. The separation takes place in the column which reside inside the column oven. The column contains the stationary phase as solid, similar to the gas chromatography. As the individual components start emerging from the column one by one, the detector detects them. A computerized recording system monitors the detector's output and provides the data according to the components which came out of the column. As each component is detected, a peak is recorded. The size of the peak is proportional to the concentration of the components. As you can see here in the picture, this mobile phase is liquid that is flowed through high pressure, and that through this injector, the compound of interest or the mixture of interest is passed in this tube, and here this pump pushes it through the stationary phase. According to the different compounds affinity to the stationary phase, they will come out of this column, and that will be detected by the detector, and also the amount of the compound will also be detected according to the number of hit on the same position that is done by these compounds. So amount that is ejected from this stationary phase will tell the amount of that particular substance in the whole of this mixture, and then the second compound, then the third compound. Likewise, a graph will be generated that will suggest the what concentration of a particular compound is there in this mixture, and whole of picture may tell the nature of the total compounds. Likewise, high pressure liquid chromatography works. Immunoassay detects or quantifies toxins or poisons in the blood or the body fluid using an immunological reaction. The importance of immunoassay method in the toxicological analysis is due to their high sensitivity and the specificity. Based on the antigen-antibody reactions, all immunoassays require the use of labeled material in order to measure the amount of antigen or antibody that is present. Labeling is required because neither antibodies nor their binding to their antigens have the high intrinsic detectability. So, a labeling is done so that they can be detected through the analyzer. An antibody is prepared against the antigen, or that is the poison or the drug, to be measured, suitably labeled, added to the unknown sample. Then labels should be such that they should be able to be detected. So then antigen antibody concentration is then measured, and that will quantify the amount of this antigen present in the mixture. For labeling, different substances may be used like the coenzymes, dry crystallates, electroactive groups, enzymes, fluorescent agents, gold, silver, selenium, colloidal particles, latex and the magnetic particles, metal chelates, oligonucleotides, radioactive elements and the stable radicals. These all can be used as a labeling agents. In the practical purpose, for the labeling, mainly radioisotopes and the enzymes are used commonly. And so the name of the technique of the immunoassay has taken the name ELISA, that is enzyme linked immunosorbent assay, and the radioimmunoassay according to the labeling done. That is, in case of ELISA, labeling is done through the enzyme, and in case of the radioimmunoassay, labeling is done through the radioisotopes. Then coming to the radioimmunoassay. Radioimmunoassay is one of the sensitive immunoassay techniques which helps in the determination of antigens or antibody in a sample with the use of radioisotopes. Radioimmunoassay was first described in 1960s for the measurement of endogenous plasma insulin by the Solomon Verson and the Rosalind Yavlo of the Veterans Administration Hospital in the New York. The classical radioimmunoassay methods are based on the principle of competitive binding. In this method, an unlabeled antigen competes with a radio-labeled antigen for the binding to an antibody with the appropriate specificity. Thus, when the mixture of radio-labeled and unlabeled antigens are incubated with the corresponding antibody, the amount of free radio-labeled antigen is directly proportional to the quantity of unlabeled antigen in the mixture. So, in this way, we can do the quantification of the drugs as well. So, how this happens can be understood in this picture, that there is the natural antibody against the particular antigen and the radio-labeled antigen for that particular antigen. Then, once all of them are mixed together, then it will join with the radio-labeled as well as the non-radio-labeled antigens. This antigen will join with the radio-labeled antibody and the non-radio-labeled antigens, and the sum of them will not be joining, as it will not belong to this particular antibody. So then the sum of the mixture will remain free, and some of them will be joined together, that will be counted, detected, and according to their activity, it may be decided that what concentration of the particular compound is there, and also what all is not present and what has been bounded with the known antibody. Like in this way we can analyze different types of the compounds, and we can decide what is the quantity also through this radioimmunoassay. So the advantages of the radioisotopes is that it is extremely sensitive. These were the very first labels used in the immunoassays. They are subsequently detected by scintillation counters. Disadvantages for the radioisotopes includes the health hazards, special attention required for the handling of the reagents. Disposal of the radioactive waste is another sort of problems. Then the training of the staffs, short half-life of the isotopes, expensive instrumentation are the certain problems associated with the radioimmunoassays. Because of the disadvantages associated with the radioisotopes, alternative non-radioactive labels like the chemiluminescent substances, enzyme, fluorescent probes, liposomes metal and metal chelates have been introduced. It is interesting to note that the antibodies against the digoxin, that is FAV, were initially developed for the radioimmunoassay technique, which permitted the correlation between the serum digoxin concentration and the clinical digoxin toxicity. It ultimately led to the realization that FAV could be used to the treatment of the digitalis poisoning. Then ELISA, enzyme labeled immunosorbent assays. Enzyme labels are the most common labels employed in immunoassay methods for the pharmaceutical compounds. Here, antibody enzyme conjugates are used. The enzyme converts an uncolored substrate to a colored product which can be quantified calorimetrically also. Advantage for the enzyme labels are that the amplification is possible, that is a single antibody enzyme immunocomplex can give rise to multiple colored detectable molecules, also known as enzyme multiplied immunoassay techniques, that is EMIT. This is also responsible for increased sensitivity of the ELISA. ELISA is a basic assay technique that is carried out to detect and measure the antibodies, hormones, peptides and the proteins in the blood. It utilizes an enzyme system to show a specific combination of an antigen with its antibody. It is a method of quantifying an antigen immobilized on a solid surface. It uses a specific antibody with a covalently coupled enzyme. The amount of antibody that binds to the antigen is proportional to the antibody of the antigen present, and which is determined spectrophotometrically by measuring the conversion of the clear substance to a colored product by the coupled enzyme. The ELISA technique was first conceptualized and developed by the Peter Perlman and Eva Engvall at Stockholm University, Sweden. The disadvantages for the ELISA is that its linear range of calorimetric detection is limited, that is, depth of color can only be up to a certain limited extent, and the color development process necessitates the additional time, steps and reagents. For overcoming this issue of the ELISA, antibody fluorophore conjugates have been developed that has the potential to eliminate this problem because the fluorescence of the immunocomplex can be measured directly on the surface or inside the solution. So, this ELISA technique or the immunoassays is useful for the poisons that are having the immunogenic properties. Like some poisons, the snake venoms are the immunogenic itself and readily amenable by the immunoassays. However, many drugs and poisons are not normally immunogenic per se due to their small molecular size. They can be made antigenic by conjugating them to a carrier protein. Then the types of immunoassays depends upon the what type of the antigens are used in the system. That can be non-competitive antigens or the competitive antigens. Depending upon that, the non-competitive is again of two types, that the one-step non-competitive immunoassays, in which the samples like blood, gastric contents suspected to contain the drug, suppose X is assumed, like the morphine or a strychnine, then one step in non-competitive immunoassay sample like the containing the blood, gastric contents suspected to contain the drug X, that may be the morphine or a strychnine, is mixed with the labeled antibody against it, that is ABX. So, the drug X binds with the labeled enzyme labeled antibody ABX and the unbound antibody is washed away. So, the bound labeled complex is measured, which is directly proportional to the amount of the drug X. So, this is the one step non-competitive immunoassay. Then the two step non-competitive immunoassays. Here the sample suspected to contain the drug X is put in a reaction tube. Antibody against the X is bound to it. Then the two-step non-competitive immunoassay. Here the sample suspected to contain the drug X is put in a reaction tube. Then the two-step non-competitive immunoassay. In this method, the already a plate or a tube containing the antibody to the drug X is bound to that plate. In that tube, this solution of the drug is added, that is sample suspected to contain the drug X is put in a reaction tube antibody against it is bound to it. X bind to the ABX, A labeled antibody is added. Then another labeled antibody is added. It binds to the antigen already bound to the ABX. The amount of the labeled antibody on the site is then measured. It is directly proportional to the concentration of the X. Since the antigen gets sandwiched between the two antibodies, it is known as the sandwich assay. Then competitive assays. They are of two types, homogeneous and heterogeneous immunoreaction, depending upon the solution in which they are placed. In the homogeneous, the solution is homogeneous. In the heterogeneous competitive assay, the solution is heterogeneous. Competitive homogeneous immunoassay, here the drug X is to be measured, then antibody against the X is prepared and bound to a solid phase as in the sandwich assays that is done in that I told you above. Same antigen X prepared in the lab with the labeled radioisotope enzymes, lab X. Then unknown sample containing the X on the lab X are added to the container having bound ABX. Lab X and X in the sample both will compete with the limited amount of the antibody X that is already attached in the tube. If the X in the sample is more, less lab X would be attached and antibody X more would remain unbound, and the vice versa may happen. The unbound lab X is measured. More unbound lab X indicates more X in the sample and the vice versa. So, in the competitive heterogeneous immunoassay, steps are the same, except in this case bound LBX is measured and more bound LBX indicates less X in the sample and the vice versa. And the likewise, we apply this immunoassay, different immunoassay techniques either on the radio immunoassay or the enzyme linked immunoassay. Similarly, you can see here in the picture, based on these types of the immunoassay, the ELISA is again of the direct ELISA, that is here the non-competitive one-step procedure. The antigen directly labeled antibody is attached with it and we are measuring the quantity of it, and then we can assess the quantification of this particular drug or the poison. Here in the indirect ELISA, the antibody complex with the antigen is being used in detection of the drug or the poison. That is why it is called indirect ELISA. Then the sandwich ELISA, as already discussed, here the already prefixed plate is there. There the antibody is fixed and the antigen will go and bind with that. Then the labeled antibody is added to it and that will be detected and that will give the quantification of the drug. Then the competitive ELISA, here the both the labeled already fixed antibody is on the plate and then the antigen will bind here and added antigen will be there, that will be labeled antigen that will compete for this antibody, and according to the contents labeled antigen that is remaining and the normal antigen that is binding with it, we can and then again a labeled antibody will be there. So based on that we can detect the different quantities and we can exactly tell the different concentration of the drug or the poison. So in this method, in this way, this ELISA technique is performed. Then the certain other chromatographic methods that are also used in certain circumstances, like calorimetry, that is based on the principle that toxins inhibits the heat production in the cell. Bacterial cells like Bacillus subtilis are taken and exposed to the sample. If the toxin is present, metabolic heat production is decreased. By the calorimetric method, the decrease in the heat production is determined. Prediction about the toxin present can be made depending upon the decrease in the metabolic heat. Then the electrochemical techniques, that is done through the potentiometry, determines the potential of the electrochemical cells, usually at zero current. The potential of the electrode of the interest respond with respect to the standard reference of electrode to changes in the concentration of the drugs. Then in the electrophoresis, the electrically charged proteinaceous drug or the poisons can be determined by this technique. In this stationary phase made up of the agar gel or the starch on the glass slide sample is placed at one end of the stationary phase. Electrodes are attached at the both end of the gel slide. As the charge flows, electrically charged drug molecule move. Gel plate is treated with a suitable cladding agent and the drug becomes visible and identified. Then similarly, in the melting point determination, identification of the drug by their melting point, which is determined by the capillary tube. This melting point is then matched with the standard tables. Then neutron activation analysis. Here, when a sample is irradiated in a nuclear reactor for a specific length of time, atoms of some element absorbs neutron and becomes a radionuclide, that is neutron activation. Radionuclides release their excess energy in the form of the gamma rays. By measuring the amount of the light absorbed, even a quantitative determination of the substance can be made. The sample must be reduced to atomic level, that is atomized for this analysis. Atomization is done by breaking the chemical bonds and releasing the individual atoms. This is done by increasing the temperature of the sample. This can be achieved, and analysis of the energy of the emissions can reveal the identity of the element originally present in the sample. This analysis is done by placing the irradiated sample into the counter system capable of detecting and recording the specific radiations, neutron activation analysis. Here you can see in the picture the incident neutron will act on the target nucleus and that will be releasing the gamma rays, and that detecting the gamma rays will help to detect the type of the particle involved. So, the disadvantage with this method is that the nuclear reactor is required, that is high cost equipment, and trained staff is also required, and the slow process because of the time required for the irradiation, cooling and the study of the emission spectra. Because of these difficulties, neutron activation analysis has now been replaced by the flameless atomic absorption spectrophotometry, FAAS mainly. Then the polarimeter measures the angle of rotation caused by the passing polarized light through a optically active substance. If a drug or a poison is optically active, it can be detected by this technique. Then the refractometry, here the under it basically identification by the refractive indices of the poisons are done. In this way, we have learned the different chromatographic techniques, we learned the spectrophotometry, and in this way we are almost able to cover the analytical aspect of toxicology. These are all techniques are applied for the purpose of the analysis of the toxins. Although theoretically this is a quite not a theoretical topic, this is a very practical topic that need to be demonstrated and the practically to be performed. So it is better to visit the toxicology lab and learn the techniques and at least see the mechanism and the practically performance that are being done in the toxicology labs. That will enrich you with a lot of knowledge related to the analytical toxicology. With this, we end the concept of the analytical toxicology. From the next class, we will cover individual poisons, and in that we will be talking details of different poisons, their fatal dose, fatal period, clinical features, their diagnosis, management, and the post-mortem changes that can be detected. Analytical aspects, if it is very important or it is specific for particular poison, then we will be talking that also. In the management part, most of the management that we already covered, and in the management that is the basic symptomatic and the syndromic approach that is followed already, and thereafter the individual poisons have a separate antidotes that will be discussed, and apart from that special points that will be required that will be covered under the individual poisons. With this, we finishes one of the complex part of the toxicology, that is analytical toxicology. Thank you so much for your patient listening. Best wishes. Thank you.